Okay, good morning, folks. This is our March EMX. I'm Bob Sinclair, and Student Yi is interviewing today. So Jane from WAS Group is very kindly running the show, as it were. And so we got two great speakers again. And the next one, the final one of the academic year, will be May 5th, 2025, in a couple of months' time. So as usual, I'll just say we've got an exciting program. I'll introduce Nigel Browning, second speaker, and I'll just pass it over to Wah to introduce Hong from UCLA. Wah, please.
Thank you very much, Bob. Good morning. It's my great pleasure this morning to introduce Hong Zhou from University of California, Los Angeles. Hong currently is a professor in the Department of Microbiology, Immunology, and Molecular Genetics. He was the founding director of the Electron Imaging Center and Nanosystems at UCLA. And he is a well-versed high-resolution single cryo-EM specialist. He was among the first in the world that demonstrate cryo-EM could solve atomic resolution structures by cryo-EM. And his interest has always been on viruses and other pathogens. And in the last few years, he actually extended himself to work on some smaller complexes, as well as develop methodology for cryotomography in the cellular context. So this morning, he's going to tell us about how his approach is being developed in this really exciting area on cryo-electron tomography. Hong, welcome to our symposium series.
Thank you so much, Wow, for the very generous introduction. And I have the next 30 minutes, so I thought I would like to share with this very, very large crowd of audience about two things. One is the methodology development we have been doing the last few years, and also tell you three stories that related to how I started in the field of cryo-EM, and then a little bit of personal story behind these three projects. So, so I actually grew up in the southern part of China. It's roughly in this area here. It's in the sort of brown and to red transition. That means a subtropical region of the region. That means that the weather is pretty hot, warm to hot. And when I grew up, one thing that bothers me a lot is mosquitoes, as shown here. And then I noticed as a child, there's a campaign going on, which actually is motivated by Chairman Mao, who happened to be my neighbor. And then I never understood it until my mother got a malaria infection. And then I understand how important that is. It turned out because of the weather and mosquitoes in that region of China, a lot of people suffer from this disease. And my mother almost died in that instance when I was a child. I think about 10 years afterwards, he really died. When I went home to say goodbye for America, I was pretty happy taking the train from the northern part of China to Hunan province, my hometown. And when I got my home, I saw that body. She was only shown here as my mother, was only 45 years old. I learned as a physicist that she died of a disease called hemorrhagic fever infection. And she died in such a tragic fashion in short time because it was given medication, aspirin, thought it was a typical flu, and then that made things worse, and then she died within a few hours. So on my flight to the America, and then I made a decision in my life. I'm going to figure out my mother died. Of course, as a physicist, I didn't know anything about it. And then I checked in the library, realized that hantavirus was actually the hemorrhagic fever infection was caused by hantavirus. And then I did some research on the internet, not a bit in the library, as before the internet age, and then saw this paper by a professor, Zhou, a medical college medicine, showing an image of virus, which is here. This is in 1987. In retrospect, this is probably the first, or among the first, if not the first, cryo image of a polymorphic virus. And then I pick a phone call. I don't know if I remember. I call you from New York City, asking whether I can become a student of you. And then I told you that I didn't know anything about biology, but I'm trained well in physics. And then you kindly offered me a summer job, and then I booked a ticket and then flew to Houston. And then I had a wonderful time learning CTF from you during that summer. For those of you who don't know what CTF stands for, it's called conscious transfer function, which is, of course, very important to determine if you want to go to high resolution. So all the years for a while, and then I have been developing methods, determine the structure, determine the methods of for determining high resolution structures. We want to actually do high resolution because in order to understand mechanisms of things, how things work, you really have to see the chemistry. So we figure out instead of just seeing how a virus look in a global fashion, you have to really understand the details. So while I'm giving the thesis project to actually push the resolution of the electron microscopy, that's way ahead of time, I have to say, in retrospect. But for those of you who don't do cryo-electron microscopy, don't do biology, I understand half of the audience will be in material science. I give this a very short introduction for you. Unlike the many modalities of imaging using electrons in materials application in biology, I'll consider the only two variations, make our life a little bit easier. The two modalities, one is called cryo-electron microscopy or cryo-EM, and the other one is called cryo-electron tomography. The tomography, ET, is shown here. T in ET stands for the same thing as CT scans T, tomography, meaning that you have to rotate the sample, and then you take multiple image and then combine them, as shown here, and you recover a 3D structure as shown here. Unfortunately, electrons are very damaging. So for biologists, that's very bad. That means you cannot have enough image before you have this image, and then your sample already damaged. So in order to go to high resolution, then that is not possible. So one way to do that is take advantage of the advantage of biology, where you can actually isolate things in multiple copies. As shown here, those viruses in isolation. You see many different viral particles. They are the same virus, but they look different because they're different orientation. So the idea is that if you have many particles of the same thing in water, then you can actually image many of those. You determine the orientation afterwards, and then put together in recovery structure as shown here. So that's by doing so, you overcome the limitation of radiation damage, and then you can get to resolution shown here, for example, 3 angstrom, and then you can determine you can see the building block of biology, namely amino acids and bases. Shown here, for example, you can look into inside the virus, you can see the genome of the virus inside this virus, which I'm going to talk about a little bit more afterwards. And the beauty of this, of course, is that you don't have to grow crystals on X-ray crystallography, where then you can image things in native form. You can see, for example, shown here, a membrane protein structure that we determined earlier on. You can look at this together with the membrane in presence of the sample, determine the structures. So these are all good, but what we biologists don't typically tell you, there are three problems. One in cryo-ET, which is very easy to do, but actually has suffered from a missing wedge problem. Namely that because you have the rotated sample, the sample is in a planar fashion, so you are missing information. There's a wedge shape that is missing. Basically you don't have information. Of course, the limited image also could have a lot of noise in the image. So you have to overcome this problem of missing wedge in order to interpret the structure properly. The second problem is what is called preferred orientation. So if you see like things like here, you make an assumption that the orientation are random, a full coverage. So basically you have all possible views so that you can build a 3D structure faithfully. That's an assumption. Typically, that's not true. If you start doing it, they actually prefer orientation in many samples. Even in biology, nowadays you do averaging, and then certain things are arranged on the membrane, for example, then they actually have preferred arrangement. And then you also have preferred orientation. You don't have uniform coverage of views. So that's the second problem. The third problem is what I call missing identification. So I mentioned to you that the beauty of cryo-EM is that you don't have to grow crystal, so you don't have to have a pure sample. So you can image everything you have. So you can see everything, but you don't know what you're looking at. You don't know. So you need to identify them before you can actually build an atomic model, because you need to have sequence information. You need to understand it's a protein, maybe it's not. Then you have to modify if it's about sugars. So you need to identify them. So those are the three problems that we typically have to solve. So we have made some contribution to each of the three problems. I'm going to show you quickly. The first one is the missing wedge problem. The software we developed is called AstroNet. It stands for Isotropic using neural network. Okay, the idea is the following. So for those that you don't do machine learning actually in 3 minutes is actually very simple. Basically, just like a Fourier transform. So you need to identify the basis function. But in the case of machine learning, you don't have that information. So you have to learn this basis function, namely this network, from your data. So typically there are two ways. One is using a ground truth. You sort of, if you know a lot of things, you have a lot of books, you can use ChatGPT to digitize all the books and learn things like that. Well, in this case, actually, we are trying to solve a structure we don't know. So, and then we don't want to make any assumption about that. So in AstroNet, we actually simulate the training data by basically taking the current distorted tomograms, reconstruction is distorted, and then you computationally rotate it, and then impose a computational missing wedge. Okay, and then you treat this doubly missing wedge tomograms here, and then try to recover the tomograms which already have some defects. So basically, we are treating the current data, which is sort of distorted, as the ground truth, and then using the simulated doubly missing wedge data is the observation. So by doing that, we learn the network, and then of course you can do that iteratively, and then have a network. Then you can apply that to your real data, and then recover missing information. All this work because the information in biology is redundant. For example, you have proteins are made of 20 amino acids. Okay, nucleic acid four bases. And that's all you need for all the biological samples in the world. So all those information actually common. They're very redundant. They're present in your sample. So, but your human eye cannot see that easily. So using by computers, you can actually train that, pick those up. So that's the philosophical point of view. If you only understand why this works. So I just want to convince you actually did work. So showing here, these are neurons growing on grids. So we're trying to do cryo-electron tomography because we only see how the neuron works. So showing here, these are synapse. So if you look at the XY plane in the tomograms, they look pretty good. It's shown here. You can see the vesicles, you can see the receptors here. But if you only look at the other two orthogonal direction, they look very, very poor. For example, the vesicle you can no longer recognize. So No, that's due to the mismatch information, a mismatch problem. So by using the arsenic reconstruction, you can see that this becomes interpretable. So simply by using the arsenic, you can have a tomogram that you can now interpret without averaging. So, for example, this is the same tomogram if you display in 3D. So this is how you can see. It's still noisy, but you can recognize big features like these carotene cages. This is important because the carotene cages are very, very important for endocytosis. They have all different shapes and geometry. So if we want to actually observe that geometry, so this allows you to do that. So that's not really help you overcome that problem. It turned out the same principle can be applied to single particles. So for those of you actually try to do cryo-EM, you all notice that this what it's called water air interface problem. Showing here, there's a grids. So basically, there's a grid, and typically scientists discover that the solid inside water, they tend to go to the surface, okay? Because the asymmetrical geometry that introduce preferential orientation, and then that present as a problem similar to that of mismatch. Basically, you don't have all you need the views. So that distribution needs to be more complicated than simple mismatch. So we can apply the same idea. We develop a software called single particle estimate. It's actually completely different, but the other principle is the same. The different because it's much more complex. So we have to basically, so using a simple network, we have to use 4 of them and try to minimize the losses. And then by doing that, you can actually overcome the preferentiation problem in single particle. And we learn actually by using these, you can even use the mission of preferentiation problem corrected reconstruction like this to actually go back to improve the overall reconstruction as shown here. So there are basically 2 modules in SPSNet. You can try it out. They actually work pretty well. Even in very severe preferentiation cases, SPSNet work. And you can also apply for single particle subtomogram averaging when you don't have all the orientations. So these are the second problem that the SPSNet allow you to overcome. The third is the identification. So one of the things you can do in cryo-EM is actually, instead of doing tomography, is simply just open the cell and have different fractions of the cell as shown here. It's shown here, illustrates the red blood cell. So you can have grow the red blood cell and then actually fractionate it and have different fractions, very crude fractions, and then you can density gradient and then put that on grid as shown here. Then you can image them, negative stain, then look at, you can see particles focus the cryo-EM is shown here. Then you can see that you have of different particles, either in solution. If you work at the membrane fraction, then you can actually see all the membrane proteins, things like that. You can also take the same sample, you do a mass spectrometry. Typically that give you like 1,000 molecules in a list as shown here. So basically you have a list of proteins in this sample. You really don't know what you're looking at. Regardless, you can use cryo-EM to determine structures. You can actually determine typically up to 10-ish structures that is most populous in the solution. And then in order to identify the structure is, then the cryo ID will pick up the best regions of the map. Density map is shown here. Typically 10 to 20 amino acids in continuation, and then you based on the shapes of the side chains, and then you build pseudocodes is shown here, and then you blast that pseudocode against the gene bank, or if you already have a mass spec list, you can blast against your list, and then identify what you have, and then you can use that sequence and then build atomic model with the cryo ID. So, so we basically did all these 3 methods, and then in the next 10 minutes or so, I'm going to show you how we apply those methods in 3 topics. Come back to my, original purpose, mainly I want to really understand how my mother died. So I, of course, continue to study viruses. So since I left WASLab, I continue at UCLA on viruses, but expanded on the scope of investigation, not only into viruses, but also other things. So shown here, getting back to the mosquitoes, which I hate so much, they actually transmit a lot of diseases, including the viruses shown here. This is a blue tongue virus, which is blue tongue because this the ships, the tongues become blue. It's a big problem in agriculture. And then this one is a dengue virus, which causes the hemorrhagic fever in the tropical region. And those are all transmitted through mosquitoes, and also the malaria I mentioned at the beginning, also transmitted bacteria by mosquitoes. So the idea is that actually they have a very complicated life. They take the mosquito bites you, and only female mosquitoes, by the way, only female mosquitoes. The bigger are female mosquitoes bite you, and then if this one carry the pathogens and then transmit to your body, and then typically go through the red blood cell shown here, and then of course then the pathogen will go through a cycle, and then you pass along the pathogen to other people, and then that's how the pandemics arise. So I'm going to show you how we use the methods we have to study viruses and also malaria, and finally the most exciting thing, mosquitoes. So I'm going to just highlight one example of what we did in each area. First, viruses. So one very important thing of interest is how virus, a very simple, I would consider it to be the simplest form of replicative machine on this planet, can use the very highly degenerate genomes they have, and then to sustain their form of life. So that they can do so because they hijack the cellular machinery. So in order to understand that process, we have to study beyond the virus. So here, these are cells cultured, infected with virus, and then those are frozen on grid. So you can see that this. You cannot see them because here you see this region of the cell. Probably those are the cells. Then in this case, we know the virus, blue tongue virus, is the RNA virus. They stay in the cellular so maybe this is the area that we observe. But these are still too thick, so we have to make it thinner, but through a focused ion beam, FIP, shown here, and then use that to make it tomographs, so shown here. So then you can use the isonet. They can interpret this directly, and then what you can do is this what I call molecular sociology of condensate. So this virus, after infection, the cell develop this condensate within the virus for the benefit of the virus. Okay, so the idea of this is that you can see that this condensate is made of both viral and cellular things. For example, they are ribosomes, obviously they are to help the virus, and then to produce the viral proteins. You'll see also this microtubule-like filaments. Those are there to transport viral particles after the virus is sampled. So using this cellular approach, then you really observe the context within the cell, how things come together to form a condensate in a way they're not too rigid, but they're not completely random. That's the concept of condensate. Because we can interpret this directly within the cellular context, then you can understand how this works. Obviously, the spatial relationship, the temporal and spatial relationship of the condensate is very, very important because for the virus to work, things should happen at the right time, in the right location, not too early, not too late. Okay, and they have to be within the confine
not bounded by membrane, so that things can live there after the virus is made. So this is how the condensate structure within the cell shows. More importantly, we can also pick up individual viral particles. These are viral intermediates. Okay, because we can recognize them, then you can average them. Okay, to improve resolution. As shown here, once you do that, then you can identify many different kind of species. Shown here are 6 of them. So we actually run out of name for those things. For example, they are called virions, that's infection particle, but there are many other things like the subcode, pre-subcode, pre-virion. We have to name these intermediates because being able to identify this allows us to look at much more detail. For example, you can zoom into details. If you can do high resolution, then you can actually see proteins, identify protein, identify RNA, in this case RNA. We actually visualize a protein that is involved in the packaging of ATPase, VP6, that's ATPase, to actually package RNA. And also we visualize the RNA-dependent RNA polymerase, which transcribe and replicate the RNA. So being able to see all these intermediate particles allows us to interpret how the virus is assembled and how they can sustain within the cell. Second example is malaria. So I mentioned that malaria is, of course, not a virus, it's more complicated. They actually is the simplest form of eukaryotic cell shown here, but they rely on they hijack the human body here, the red blood cell, for their purpose, for their benefit. In other words, they take advantage. They parasite basically. They parasite and they use the human cells for their benefit. So one of the questions is to understand how they do that, right? So in order to understand that, then it's important to look at the relative environment. So using the recombinant approach and purify, we want to look at the relative context. So by breaking the cell, and then you can actually image the cellular extracts as shown here. You can do a 2-dimensional classification. You see all different kind of particles. And then some familiar things appear, for example, the proteasome. There's other things that have symmetry emerge. And then I want to highlight this last row here. These are asymmetrical features, pretty big. And then you can combine them and get a structure down here. Looks pretty good. It's 3.7. So you can see bumps, things like this. You can see helices, but you don't know what they are. So the ID come to play here. So you can actually zoom into regions of the protein, and then here pick out the regions that is resolved the best. So you can identify them. By doing that, Mimi Ho, who was a student here, then he became professor at Columbia now. So identify actually is made of 3 proteins: Rho PH2, Plaque 3.1, and Rho PH3. So more importantly, within this protein shown here, the Plaque 3.1, we saw a helix buried inside the protein. This is of particular interest because this protein was supposed, based on the sequence, to be a membrane protein, with this helix actually suggested to be transmembrane helix anchor to the host the red blood cell membrane. But it turned out here is actually no, and then the sample itself is in the solution, so it's soluble, so it doesn't make sense. And then when you examine this more carefully, you see that this actually has a lot of disulfide bond holding this inside. Okay, and then we studied in the literature and realized there are 2 papers in the literature. One is actually published, the both actually published about 20 years ago, and then talk about this protein being actually forming a channel for the benefit of the box within the red blood cell. And then this disulfide bond of the helium helix, it makes total sense because what happens is really the protein needs to go through multiple environments. So we think that this disulfide bond is actually a trigger that's used for the box to sense the change in environment so that it can be released. The helix can be released at the right time in the right place, like here, to be inserted into the membrane of the red blood cell, and then that becomes a channel. Last 1 minute, so I'm going to talk about mosquito. So
So we demonstrate you can do all that. So, and then I have this crazy idea. I said, why don't you just pick up a mosquito and image that? Of course, that's too big. So we take the simple thing, is that because this mosquito, when you take a bite, it's the saliva fluid that actually causes the problem. So we actually take that and image that. So if you do that, this is what you see. This is a negative stain image on the crowd EM. You see actually pretty impressive, right? It look pretty impressive to me when I saw this, because you see the particles. And then we pick out the biggest one here, and then did a structure shown here. Interestingly, we identified this is also membrane protein, SGS1, okay? And then it has actually particularly transmembrane domain TMD here, but that domain is actually hidden also in this cavity inside this big protein, actually it's over 2,000 amino acids. So we think that this is similar to the channel protein of the bug that also have multi-stage of the protein. And it makes sense because the saliva need to be injected into our body, and then that need to change during that process. So with that, I'm going to just finish my presentation. So basically tell you, hopefully you are convinced that probably EM now is pretty powerful. I highlight three things we did to contribute to the field. One is SNET that is developed to overcome the mismatch problem, and the single particle SNET that overcome computationally the anisotropy orientation problem, and Cloud ID allow you to identify body image in 3D. And then I highlight three examples, starting from virus, Bruton virus, malaria cell, and also the mosquito that transmit those mechanisms. So with that, I'm going to acknowledge people who did the work. Winter, as a postdoc in lab, developed the methods, and then Mimi, who is now at Columbia, who contribute all the most the malaria-related and Cloud ID program, together with other students, Jason and Sharin Lee in the group, and Shihan and Shan, and did the work on the mosquito. And I have a list of collaborators. In the interest of time, I'm going to go through them. Thank you so much for your attention.
Wow, that's very impressive, Hong, with all these development last few years. Congratulations. Okay, I think the floor is now open to questions.
Bob? I cannot hear you, Bob.
Yes, thank you. I had a question, Hong, about you're doing this beautiful tomography and also some great work on these viruses. What is a typical dose rate which you do? How much can you get away with the cryo approach for these samples, and how well do they hold up?
Well, it's actually sample dependent, but we typically the max we go to is about 100 electrons per angstrom square. But typically you want to retain things within 60 electrons.
That still sounds pretty low, pretty small.
Oh, yeah. Then you fry things when you increase. And that's why the averaging is essential in order to put resolution.
Do you lose any information through that averaging?
Yeah, of course. Anything that differ from each other, it will be smeared. The beauty of biology is that you can image, if you have like, wow, you can have 5 talent grills in a national center, then you can dedicate one then to image mosquitoes. Then you can solve all the structures.
Hong, you're wrong. I cannot do that. These grills are for the community. I wish I could dedicate one for my own project.
Yeah, I wish I could do that too.
On the record, it cannot be done and it will not be done for transparency reason. Okay, right. Let's ask another question from the audience. Stephen Oliver, who asks if any evidence of asymmetry in different types of reconstructions of your Bluetongue virus?
Of course, the genome inside is asymmetric, and we have to relax that in order to recover the polymerase complex. Outside of that, we did some experiment, try to figure out how virus entry. We didn't have time to show, but during the process, the interaction of the virus began to actually change, and then we can visualize the membrane insertion, and of course during that process, that is very asymmetric also. Asymmetry is a great thing, and then we have to make sure that we can recover that if you want to see dynamic structures.
How far are you from capturing the high resolution structure of that that process?
Right now we are in the range of about 20 angstroms. I always tell my students there's no limit, and the physicists will tell you that you can image subatomic details. So we are in a lucky situation in biology. We don't need to do that. So, but just a matter of, it's you refer to that we need a lot of particles, and then we don't actually have enough resource to do that. It's just a matter of we need to average a lot of things. We don't have enough money to cover all the cost.
All right, good. Another question is, how do you validate the features you retrieve from the missing words by your isolate method?
So a fantastic question. The biggest concern for any machine learning program is that sort of what's called overfitting. So one of the things we are very aware of that. We try to do everything possible to try to alleviate that kind of concern. One of the things we never use any models to train a neural network. So everything arises from the data. Okay, but still, sometimes you could create artificial noise. Okay, that may manifest in structures. So one way to overcome that, to validate, is actually take advantage of the reduced gradient of the sequence in biology, which are all free and very cheap to get if you don't have one. So you have all the amino acid sequence. Then if the model you see through that process match your independently determined sequence, and that I would consider to be the best validation that we biology have.
Okay. Another questions related to artifacts. Can you easily distinguish of the features of interest of the sample prepared in Lamella for tomography, and how do you ensure that artifacts can be avoided?
I guess Lamella, I guess you're talking about low resolution interpretation for cellular tomography. I actually don't have actually good answer for that. For many years, personally, I tried to avoid doing tomography, partially have concern about overinterpretation of low resolution data. For those that you know me, I actually mainly do high resolution in the last 20 years or so for that concern. So I fully, right now I don't have a good way to do that. I always try to say that push the resolution. Once you reach about 3 or 4 angstrom resolution, then you could use the sequence as the independent check. I guess nowadays you can take advantage of AlphaFold that help you to look at the tomograms even at low resolution. So if you match the secondary structures like 8 or 9 angstrom resolution, if the fold matches, there's a fold independent from AlphaFold prediction. I think that's pretty good validation in cellular tomography data.
Okay, good. Okay, Katie has a question.
Yeah, thank you for the great talk. I really appreciated it. I was curious, kind of a similar question with your ID, like identification. Are there ever some redundancies, like two structures that could give you a very similar output? And if so, how do you distinguish between them?
Yeah, actually, their structures have very similar features. For example, you can have isoforms of protein. They only differ like 1% in sequence. Actually, they actually in tissues. Actually, this is important. The tissue specification. If you look at the human body, we only have 23,000 genes, but actually we do a lot of things, right? Those isoforms are very important. So if you do have that kind of concern, which you should pay attention, then the great thing is that we now, once we have a structure, now we have the sequence, you can just go back to the old, old approach, namely the expressive protein, and then just quickly do that kind of structures to validate it. And also, the performance approach, of course, just image more, so improve resolution. So once you reach about 2 angstrom, you can actually differentiate different kind of amino acids for sure. So that should be that's another way to do it.
Thanks.
Okay, another question related to cryo ID. The question is one of the wish in the past was to use the microscope to sequence the DNA. Do you think your cryo ID is a viable approach toward that goal?
Certainly you can do it nowadays, but I don't advocate it because that's a foolish way of doing it. UCLA, I drop a DNA sample, and then they give me the sequence back in the second day. I paid $10, right? So that's a great technology. You should take advantage of it. So sequencing information is so cheap.
Okay, that's a good answer. Okay, there's another questions about your isolate protocol. What resolution you think you get in your restored regions of the data through the isolate?
Not too high. It's typically in the 30 angstrom range. And then the good news is that once you actually can see it, you can actually you can pick particles and average improved resolution. Bob, are you talking? We cannot hear you if you're talking. No.
No. Okay, let me ask another last question is, cryo-EM is a kind of expensive. So do you see any way to cut the cost so the less privileged individual like place like UCLA can do it also?
Well, yeah, sure, absolutely. The pioneers like Wow and others in the field, they thought about this even 10, 15 years ago. That's why the idea behind the National User Facility. Even me, as a founding director of the UCLA cryo-EM, I cannot resist that temptation to use their facility simply because they don't ask me to pay anything. Well, at UCLA, I had to pay $1,700 a day. So take advantage of that. Actually, they'll give you more easily if you can demonstrate you don't have access locally, right? Well, yeah.
That's the greatest idea you present today. Yeah, please come to use our facility, and I actually love it. And now let's ask the last questions. Okay, and then if you only have a single wheel and want to use isolate, would you suggest collecting at one additional tube or two?
Oh, yeah, absolute question. Yeah. So single view is obviously not enough. Okay, so you do need to because it's somebody you need to have different views. In the paper we actually talk about that. So you can actually having a simple tilt at 30 degrees is a great thing to do. It's easy to do. A great thing to do. That will simplify the problem.
Okay, so the question raised by Michael Album, I think I read it wrongly. What he meant is to determine the DNA sequences in association with the protein structures inside the virus.
Oh, virus, yeah. Yeah, we haven't resolved the DNA stuff very hard resolution. We have we take it to Watson Crick model to model our DNA, but in reality, we don't see the basis for the most part of DNA. So we can see a few in regions that are attached to protein. So yeah, the answer is probably no.
Okay, then I think in view of time, and I would pass the forum to Bob to introduce our next speaker. Hong, thank you very much.
Thank you.
Wow, fantastic, Hong. Really appreciate that presentation, and keep working to get rid of that mosquito problem. Anyway, it gives me a great pleasure to move on to our second speaker, Professor Nigel Browning, who's now director of the Albert Crew Center for Scanning TEM at the University of Liverpool, also professor of materials design, manufacturing and engineering. Certainly, Nigel is well known in the United States. He had positions at Oak Ridge National Lab and University of Illinois, Chicago, UC Davis, and most recently at Pacific Northwest National Lab. But he came back to UK and is now setting up a very exciting relativistic, ultra-fast electron diffraction and imaging, RUDI, which he'll tell us about. And he'll also be the chair of the next International Microscopy Congress in 2026 in Liverpool, which is my hometown. So that's exciting for me as well. So I don't think I can say any more, Nigel, but I'll leave it to you to tell us what's going on there in Liverpool and to tell us what's going to happen in the future as well. And also your journey through ultra-fast electron microscopy. Nigel, please.
Okay, well, thank you very much, Bob. Let me just see if I can find the presentation to share. Can everyone see that? Is that working now? Yep. Okay, good. All right, well, thank you very much. So what I'm going to do today is really talk about what we're doing to build RUDI, or the Relativistic Ultrafast Electron Diffraction and Imaging facility. Now, I must say up front, this is going to be completely different in style to Hong's talk. You'll get none of the wonderful results and interpretations coming out of it. What I'm going to talk about is really aspirational goals of what we're trying to get from the design of a new microscope. So first off, what actually is RUDI and what we're going to try and do with it? Well, the idea is that it will be a national user facility, and it will use MEV electrons to get to ultrafast timescales. And the goal is to try and combine what is the state of the art in electron diffraction, MEV accelerator design, lenses, stages, detectors. And you'll see in the little images I've got on the right-hand side here, the one at the top, I think, is the SLAC UED system. And if we can put them all together and to take the advances that have come over the last few years in all of these areas, then we should be able to get something that will allow us to get really transformational insights into a lot of different science themes. And the 5 that we're working on are chemical change, materials and extremes, quantum, energy, and biosciences. And I'll talk briefly as I go through about what we're trying to do in each of those areas. So this is aimed to be a national facility, and to get it to operate, we're going to leverage some of the facilities that are already in the UK. The UK, as a small country, likes to work together on things and have all of the overlap between existing institutes that are already set up. So the main partners in this are the University of Liverpool, and there we have the Albert Crew Center for Electron Microscopy. And what this picture is the Materials Innovation Factory. And this is basically an approach to using artificial intelligence and robotics for high throughput material synthesis. And I'll talk a little bit about how that will play into the RUDI project as we go forward. We then have the Rosalind Franklin Institute. This is an institute that's designed to use advances in material science, characterization methods to advance structural biology. And then we have SDFC Daresbury, which is one of the 2 national labs in the UK. And here we have the Cockcroft Institute, which is really geared towards the design of accelerators. And so combining all this together, we have accelerators, diffraction, imaging, and scientific applications. Now, before I talk about how we're going to move forward with RUDI, it's important really to understand the types of experiments that we can do inside of an ultrafast system. So on the left-hand side here, we have something that we call stroboscopic approach. And the idea behind this is it's more of a pump probe type of experiment, where you go through the process and each pulse has maybe 1 to 100 electrons in it. So to create an image, which you'll need 10 to the 8 to 10 to the 9 electrons to get that image, what you have to do is to have obviously millions to billions of different shots, and you pump and you probe and you pump and you probe, and you go through that process. Now, for stroboscopic to work, then you need to have something which is completely reversible, because you're going to have to do millions of different excitations to the sample. So there you're going to lose things in terms of spatial resolution. What you're going to be able to do is to have something that obviously has to be reversible, because it has to go back to the original ground state to be excited again. So there's a range of experiments that you can do under the reversible side of things. On the right-hand side of the slide, there is what we call single shot or single shot movie mode, or something where we can get multiple shots to look at the progression of dynamic interactions. And here the idea is that you really need to have enough electrons in one pulse. And if you can do that, you can look at the whole image from a single pulse, and that will allow you to study things that are irreversible. So pretty much all of material science, when you're looking at what happens to the structures under deformation processes, nucleation and growth, when you're looking at biology, catalysis, materials under extreme conditions, those are the things where we have to go to the single shot approach. And those two sides of the process are things that inform the way that we're going to design the instrument. The other part of it is that there is also a difference between how you would want to do diffraction and how you would want to do imaging. There are quite a few instruments in the world that allow us to do ultra-fast electron diffraction at megavolt type of energies. And here the idea behind going to MEV is the fact that the relativity helps you. And so you can get shorter pulses, you can combine the electrons together, and you can get faster time resolution that comes out of it. On the imaging side, there is a lot of work that's done on what is called an ultra-fast electron microscope. This is a low voltage, and by low voltage, we're talking about 1 to 200 kV, facilities where you can do stroboscopic experiments. This is an approach that was pioneered by Ahmed Zhou at Caltech, and there are plenty of examples of this around the world where you can see UEM facilities doing stroboscopic type experiments. So we get imaging by stroboscopic, we get ultra-fast diffraction at high voltages, and this leads directly into what it is about RUDI that is going to be unique on a global scale. What we're aiming to do to get out of this is to basically be able to get the whole process done under single shots. So we have to get enough information or enough electrons into our pulses to be able to study very fast dynamics that is irreversible. If we go into a microscope, we know that in electron microscopy, there's a lot of work been done over the last 30 odd years in operando conditions, whether it's gases, liquids, mechanical deformation, optical, whether you want to go through the process and look at cryo conditions, all of that can be combined. And as long as we get the ability to get single shots, then we'll be able to do transformational type irreversible reactions. In terms of the process of how it will be arranged as a facility, the goal is to start up front with a hub and spoke national facility. And here, what we're talking about is essentially a data-driven instrument design and user program, and I'll go through a little bit of what goes into that. And then we have hybrid interactions in terms of it's unlikely that anyone will actually come to use the facility in Daresbury, where it's going to be set up. And that's because the machine is going to sit in a bunker, and it's in a bunker because you can't shield electrons efficiently at 4 MeV, and so therefore no one really is going to be sitting on the microscope. And if no one's sitting on the microscope, you really should just send samples and run it remotely. And if all of this comes together, then we should be able to get around 10 femtoseconds in diffraction. And the goal for the imaging line is to get to sub-nanometers and about 1 picosecond of temporal resolution. Okay, so we have initial designs for this. This is an idea of the schematic. Obviously, the process is going to be slightly different in what you would understand from an electron microscope. The scale bar here, you can see is about 15 meters in length. And the key is that this will not be a vertical microscope, it will be a horizontal microscope. So this thing will be laid out like a UED system, an MEV UED system. Now that creates all kinds of challenges for engineering of a microscope, not least of which, if you understand how you put a specimen into a microscope, there's a difference if that's going to be put in horizontally or if you're going to have to put it in vertically if you have a microscope that's horizontal. So all of these are design considerations that have to come through. The whole process will be laid out with two essentially side-by-side instruments, and the reason for that is so that we can have all of the laser controls going into multiple systems, and the laser controls are the things that give us the optical excitation and the temporal resolution that will come out of the process. But if we take the range of temporal resolution that we can get out of this, here is essentially where Rudy will cover. It's somewhere between a microsecond and a femtosecond, and on the bottom end, it fits in with the kinds of experiments that you can do out of a conventional electron microscope, and at the highest end of temporal resolution, you get into attosecond measurements. But Rudy will sit right in the middle of this, and so you have all kinds of electronic, atomic, and spin degrees of freedom for real fundamental type phenomena that you can actually begin to examine using this capability. So there are 5 main science themes that we're working on in this process, and here I'll just go through them very briefly. On the top left-hand side, we have chemical change, and the idea behind this is really hardcore ultra-fast science, looking at what molecular interactions are, how they change between gases, liquids, and solids, and how we understand the interaction of the first process in controlling a reaction as it takes place. In the middle, we have materials under extreme conditions. The advantage of going to MEV systems is we can have thicker samples. If we have thicker samples, we can do mechanical deformations, we can get laser shocks into the system, we can study advanced processing techniques for material science. There are all kinds of energetic interactions with materials that you can study, deformation materials under earthquake type environments. We can create plasmas inside of the microscope and look at plasma physics and how plasmas change under different controls. So there's all the extreme conditions that go into it. On the top right, we have quantum materials and processes. This is something, again, on the ultra-fast timescale. We're going to use diffraction. We're going to look at things like electronic phase transitions, how electronic phase transitions change around interfaces and defects, and get through to that whole range of phenomena. At the bottom, on the left, we have energy systems. This is a picture of a battery where you have the solid electrolyte interface that's formed between a solid electrolyte and a liquid electrolyte. And here, to get the resolution to be able to see the process as it's taking place, you need to be on the nanometer to micron scale, and you need to have the time resolution to see individual interactions, and we'll be able to do that. We'll also be able to put real battery systems into the microscope because we'll have the ability to look at thicker samples. For catalysis, we can go through and do dynamics, but we can now do dynamics when we can go to higher pressures. So typically when you do catalysis inside of an electron microscope, you can get up to 1 atmosphere of pressure if you're lucky, and not very high temperatures. Inside of Rudy, we should be able to get up to above 10 atmospheres of pressure and whatever temperature we want, because we're only getting that temperature for a very short period of time. So this helps to avoid the pressure gap that exists for catalysis science. And then because we have optical excitations, we can do photocatalysis, and we can do solar systems as well. And then the final one is the biosciences, and this is sort of going back to more traditional biology of going to higher voltages. If we go to higher voltages, we can look at thicker samples, we can get whole cells inside of the microscope, and we can look at the dynamics that process in that. So altogether, we have a wide range of science interactions that are going to be taking place. Now, in terms of building Rudy, and at the moment, the construction is a 7-year project that's supposed to start at the end of this year, the beginning of next year, and supposed to is because it's a government process, and we all know what's going on in the world at the moment, and it's not really very stable. So we assume that we're going to get the funding for this, and we're moving forward with it. As we're building the system, we can build whatever stages we want into the process. And the 2 lines are really focused towards different types of science. So we have in situ stages for the electron microscope, which is going to be the imaging line. The difference in building those is we're going to have much more flexibility over high resolution microscopes because we have a larger gap in our pole piece. We can look at thicker samples. It makes engineering a lot easier. For the diffraction line, we can be a little bit more creative. So one of the things that will be developed for the quantum line is essentially to have a dilution fridge inside of the line. There we can get to 5 millikelvin of temperature, and we can look at diffraction or phase transitions inside of quantum materials using this approach. We can also have liquid jets, gases, all of those kinds of things in diffraction line as well. And the advantage of separating them is the fact that effectively for a diffraction line, you have almost unlimited space for the stages that you want to put into the system. Another part of the process is that having an MEV system means that we have a very specific type of experiment that we can do. But that doesn't count for all of the ultrafast that you would like to do. And in a minute, I'll talk a little bit about where some of the compressed sensing, where some of the artificial intelligence feeds into that. But one of the processes is you'd also like to have a lot of other ultrafast experiments where you can filter in to Rudy. So we'll have a basic UEM microscope. We'll talk about low energy electron diffraction, low energy electron microscopy, and then also regular voltage ultrafast electron diffraction, and essentially X-ray diffraction as well under those conditions. And the idea behind this is that we will need to do fundamental characterization of materials and processes before they go into the Rudy system. And because the Rudy system is in a bunker, as I said before, effectively these are the onboarding ramps for running experiments at Rudy. Now, as with everything in the modern world, if you run into something which is a bit of a challenge, you just say that AI will solve the problem for you. And so we're going to also use AI in this process. The way that we're going to use AI to help us with Rudy goes into 3 major areas. One is to construct a digital twin. Again, the access to Rudy is going to be difficult. It's going to be very time-consuming and very expensive. And so anything that we can do to help that with a digital twin is something that we'll be developing. We'll have compressive sensing, and I'll talk about that in a minute, and where we get into what we call image-free hypothesis testing, which is basically that the image is just the waypoint to the analysis. And so if you're going to get an image and you're going to have a digital twin, then actually we want to do hypothesis testing rather than just getting an image. The other part of it is that we're going to use effectively data science. Typically when people build facilities, they run with the concept that if we build it, people will come and use it. They'll recognize the quality of the facility, the unique capabilities, and what you can do with it. Well, that's true in some cases where you have a certain set of people that are already working at the forefront of science in that particular area. However, it doesn't mean that we shouldn't be looking for different applications that we can use with the system. So a big part of the Rudy artificial intelligence structure is actually to go out and use data science to find experiments that could be using the facility. And so this is really to just basically scour through the literature, find experiments where you should actually use Rudy to go through that process. So all of that together means that as well as the physical infrastructure, we also have the computational infrastructure as well being set up for Rudy. Okay, now why do we want to do that for something like Rudy? Now, as I said before, diffraction is going to be on a stroboscopic scale. There's lots of facilities out there to do that. What you're really talking about is timing precision in terms of the way you want to deal with it. The imaging side is far more complicated. And the reason for that is because we have to squish the electrons together to make a pulse. And if we do that, because they're negatively charged, they'll push each other apart. So effectively, that creates a problem for imaging. And so what we can do is deal with the way these problems come in a lot of cases, because overall, you have expansion in the XY plane, which is just a blurring, which you can correct for. It's just lateral space charge. We have a time dilation in the direction of the propagation, which is basically electrons spreading out in that direction. The thing that we can't correct for are things that are related to the trajectory of the electron. And the reason this is important is because when electrons go through the sample, you get changes in momentum of those electrons, which is caused by the interaction with the sample. Now, if all those electrons are bouncing off each other at the same time, what that's going to be doing is blurring out those interactions. And so we have to control the electron beam in a way that allows us to reduce the amount of electrons we use in our system. So to make that work, what we're going to do is a form of artificial intelligence called sparse dictionary learning. And the way that this works is that if we look at a complex signal like we have on the right-hand side here, that signal can be made up of dictionary elements, whether those are sines or cosines or whatever they happen to be. Now, if you know what the dictionary elements are, it turns out that you actually don't need very much of the data in your measured signal to be able to fit to that dictionary. And so you can actually get down to very close to about 1% of the data before you start to notice differences in the system. So what that means is actually we can get down to only requiring about 1% of the data that you would normally use in an image. So if we can set the system up to do this, we should be able to get rid of some of the electrons. Now, this is easily extendable to 2 dimensions. So this is the way that JPEG compression works. The difference that we're doing in the microscope and the way that we're applying the artificial intelligence is not as a processing after we acquire the images, but setting it up so that when we take the images, we're actually getting the minimum amount of data coming out of our image, which we can then reconstruct fully to get our image. Now, this works well on conventional images. Here is just a couple of examples. 2 images. It works on hyperspectral image. You can see on the color image on the left-hand side of a castle in France, and then the Barbara image on the right-hand side. You can see that effectively by using very small amounts of data and using the dictionary, we can reconstruct the image very well. Now, this works well on experimentally for microscopes. It's been demonstrated on high-resolution images. And here you can see just a reconstruction of silicon, taking about 20% of the data. Unlike artificial intelligence that requires training data, because we're implementing this on each individual image, if you change the image, you can change the reconstruction as well. So we can run it very quickly. You can see that changing focus doesn't do anything, changing magnification doesn't do anything, and you can acquire the data sets. The other big advantage of this is you can correct for problems that you have in your data set. And here again, there's a live image on the top side. What you can see in the reconstruction of the silicon dumbbells is that you actually have either there's a slight defocus, there's a slight tilt, or there's a slight astigmatism, and you don't resolve the silicon dumbbells. If you use a simulated image as the training set or the reconstruction set that goes into it, then effectively you can reconstruct a high resolution image. Now, what this means for Rudy is, of course, that we can take high resolution images that are required from our ecosystem around Rudy and use those to create dictionaries that allow us to interpret our image. This works very, very well in terms of high resolution microscopy. Here's another example from an SEM. On the top is a backscattered image. On the bottom is an aluminum map from EDS. And you can see in the zoomed in image in the middle, it looks like we're missing data. We're not missing data because we didn't acquire it. We're missing data because the scattering cross-section wasn't high enough, the dwell time wasn't long enough, and so we missed data. But if we take our dictionary from our backscattered electron image and we recreate from our EDS map, which you see here, you end up getting an image which is much higher resolution because the dictionary will help you. And so effectively, the process that's involved in the way that we're going to run Rudy that will allow us to get images at the highest temporal and spatial resolution is to use images from high resolution microscopes that are static, use images from ultra-fast techniques that are not at the same scale as Rudy, create our dictionaries and put that into our imaging system, which will allow us to get faster and higher spatial resolution images coming out. And combining all of those things together, the advances of the microscope with the advances of the imaging gives us a way of being able to image on these very fast time and spatial resolution scales. The other part, which I said I'd mention at the start, which I don't have a slide for, but I'll just talk a little bit about, is that when you think about where robotics and artificial intelligence and high throughput synthesis of materials is concerned, it's much easier to change the material you're studying than it is to change the microscope. The microscope is very expensive. Really is going to be 125 million pounds or 150 million dollars to get the whole thing built. If we can't have the spatial and temporal resolution to see something, it's much better to redesign the material so that you change the dynamics and the spatial resolution you need to be able to see something than it is to build a new Rudy. And so the high throughput is going to be linked to all of this so that we can control the images and the microscope and the samples that are going in to the fundamental information that we come out. And if we do that, we can basically get the whole thing to work. Here's an example of just a low resolution TEM image taken. This is actually data from Peter Crozier. In this CMOS device, you only have about 20% of the pixels that have electron scattering in them, and you can see that you can reconstruct a very high resolution image that comes out from that. So finally, I'd like to show an example of why we were going through all of this example of or this hard work to try and get an image. This is an example of the DTEM that we built at Lawrence Livermore National Lab, good 15, 20 years ago now. The idea behind it was to try and do some of these high resolution images. This was using a laser to create a photoemission pulse, and that pulse was then synced to stimulating the specimen, and then you would basically be able to get high resolution. The example I want to show is studying something which is called a reactive multilayer foil. This, in this case, were basically nano layers of nickel and aluminium. When you put either a spark or heat on one end of this foil, the nickel and the aluminum mix, forms an intermetallic, and that reaction zone is exothermic, and so it is self-propagating. It propagates at the speed of about 10 meters per second, which means that in terms of nanoscale, you need to be able to see 10 nanometers per nanosecond to be able to study that reaction. Now, we could do that inside of the DTEM, and here are the images of the interface. This is a 200 kV microscope, so we don't get the temporal resolution that we were hoping to get, or we will hope to get out of Rudy. We also don't get the spatial resolution that we get out of Rudy. You can see it's microns on the scale bar here. But even without that temporal and spatial resolution, you can see the reaction front here, and you can see these squiggly lines that come back from the reaction front, and they terminate here at some point after the reaction front propagates. So the question is, what is this squiggly line? Even with the temporal resolution that we had on the DTEM there is no way that you can quench this reaction quick enough to be able to see these effects taking place. Well, if we study different compositions, so we go to an aluminum-rich one, here with aluminum-rich, you again see the squiggly lines, but they don't exist for as long. And if you go to a stoichiometric mixture of nickel and aluminum, you end up getting something without squiggly lines. Now, if we go to the phase diagram and look at what could be happening, in this case, I think this shows why you would want to do the images, because actually when you go to the phase diagram, it's obvious what's going on. We heat the system to about 1500 degrees inside of the microscope. That's what the laser does. And if you go across and you go to the position where you're a little bit aluminum rich, and you go to the bit where you're a little bit nickel rich, you can see you're in a two-phase regime. If you're stoichiometric, then you're already in the intermetallic phase. So effectively, that reaction, when it's stoichiometric, is a solid state reaction. And when it's non-stoichiometric, you have a liquid that is present. And it makes a lot of sense to see that when you look at the phase diagram. But the reality is before we could see that inside of the microscope, nobody chose to discuss it as a mixed phase reaction. And this is really the power, or the reason why we're going through all the process of creating Rudy. It's so that we can see these unexpected things. You get an image, and then you can do the science, and you can interpret what's going on. So where we're at the moment is that we are developing the applications. The construction of Rudy is a 7-year project. Some of that is political, some of that is government, some of that is just the time to design and build a system like this. If anyone is interested, we're happy to hear from you about the potential applications. We have all kinds of contact addresses to get into that. We're happy to register anyone who's interested in being involved with that. And we'd love to have ideas from people about how to design the experiment. At the moment, because we haven't built it yet, designing an experiment is free. We can design, we have 125 million pounds to build it. What we choose to buy in terms of the specimen stages and the applications at this point is not decided. So before it's actually purchased, it doesn't really cost anything different to change that to make something that will work for anyone's idea of what will be done. And finally, as Bob mentioned in the introduction, I'd like to invite everyone to Liverpool. Liverpool is quite an old city in the UK. It was founded by King John, and he is that King John from the Robin Hood legends. And so the city itself was the richest city in the world in the 1700s, and we still have these traditional docks that are there, along with very modern buildings that are around there as well. and the International Microscopy Congress is going to be in 2026. It is actually in what is usually the nicest week for weather in the UK, but I have to say that's not usually as nice as the nicest week for weather in California. So as long as everyone is prepared for a little bit of rain, we'd love to see you here in Liverpool for the IMC meeting. And with that, I shall finish, and I shall be happy to take any questions that you may have.
Wonderful. Thank you very much, Nigel, for that quick overview of what you're doing. I just want to find out, where will this microscope be built? Certainly, so being horizontal is perhaps not so unexpected because of the length, and its beam lines are normally horizontal, so it seems natural. Where will it actually be built, and are there going to be any stability environmental issues which you have to take care of, or just it's because it's high voltage, then it's not a problem anymore? I don't know.
Yeah, so this picture that we have here, this is actually the accelerator tower for the synchrotron in the UK, which used to be at Daresbury, which is about 6 miles outside of Liverpool. In about 20 odd years ago, the decision was made to move the national synchrotron in the UK down to Harwell, which is the second national lab in the UK, which is just outside of Oxford. So essentially, in this hall that exists here was all of the accelerator, the electron accelerator processes for a synchrotron. And so when the synchrotron moved, that hall is open, and so we are taking space in that accelerator hall for Rudy to go into that. So there's already the space. It's already on a national lab. It will just go into the space that was vacated when the synchrotron moved out.
And the super stem is there as well, so that's a huge advantage as well.
Yeah, so on this picture here, the super stem is over here on the right hand side behind the trees. So effectively, the idea behind it is that Daresbury will have both the highest spatial resolution stem in the super stem, and then the highest temporal resolution microscope as well, and that will form a new, effectively national facility in the UK, which has both high spatial resolution, and of course the superstem focuses a lot on or energy resolution as well. So highest spatial, highest energy, and then highest temporal resolution for imaging and diffraction in the world.
And for people who are not so familiar with the geography of the UK, it's about halfway between the great northern cities of Liverpool and Manchester. Although one would hope that the Manchester soccer fans don't come at the same time as the Liverpool soccer fans.
Well, yes. Well, there's not as much trouble at the moment, of course, because Manchester United are not very good at the moment. Anyway.
Well, we'll leave that one aside. So, Kate, you have a question?
Yeah, thank you very much, Nigel. This sounds like it's going to be an incredible program. I'm curious, or questions that I think a lot about are kind of nucleation and growth, or those initial nucleation and growth steps of materials. I'm curious, do you have any plans to integrate kind of deposition sources or like methods for doing growth inside some of these in situ stages?
Yeah, so we will have, obviously, so the advantage of Rudy with the bigger gap is for things like liquid cells, gas cells, you have much more space to deal with things. Another question that usually comes up in worrying about those things is, what is the effect of the beam going to be if you're going to try and study those things? It turns out going to higher voltages is actually better for all of these studies, because most of the problems with studying nucleation and growth or studying beam sensitive materials are related to ionization damage, because you're trying to really change the chemistry or control the chemistry when you do that. When you go to higher voltages, you have less ionization damage, and so effectively most of the beam goes straight through. So we don't have plans at the moment to have like an atom source or something to create clusters in the beam. We will have liquid and gas stages where we can put things in and we can look at nucleation and growth experiments under those conditions. We are going to have an ion source so that we can look at radiation effects. So you can basically just implant ions and look at the temporal cascade that takes place under those conditions. But at the moment, we haven't thought about atom sources. If you have some ideas of that, or if you know how much it would cost to implement it, I'd love to hear about it, because, as I said, at this point, nothing has been made or built or put in, and so we can change it.
I see. And you mentioned gas cells, so you're planning to do most of the gas studies in a cell environment and not have kind of gas flow directly into the area.
So there are two sides that we will do on this. So for the diffraction line, we're likely to do gas flow because we're not worried so much about spatial resolution in that, and so it will be very similar to what you would have in an exfil or a synchrotron, where you'd have a gas flow or a liquid jet source to go through it. For the microscope, because we can go to thicker cells, we can have essentially thicker windows, which means we can go to higher pressures. So effectively, we should be able to go up above 10 atmospheres of pressure, have that sustainable in a cell, and then having a cell is better than having the flow. And so the idea the flow would be on the diffraction line, the cell would be on the imaging line, where we can look at higher pressures or higher flow rates if you're looking at liquids. You can look at different pressures in terms of speed of flow as well, when you have those kinds of cells. And so there's a mixture of both, depending on what is the requirement for the experiment, whether you need spatial resolution, whether you need temporal resolution, those kinds of things.
I see. Very interesting. Very, very cool system. Thanks.
Thanks, Kate. So let's go to the Q&A. I'm not sure if the Marin's question is for Hong or for Nigel, so maybe Wak can help look. But going on to Kirk McGregor's question, can the quantum materials and processes thrust encompass quantum sensing and electron entanglement experiments?
So what we can do is to get cold enough to be able to think about those kinds of experiments. What we are aiming to do over the next seven years, because 7 years to design and build is a blessing and a curse. It's a curse that it's 7 years until you have to build it, but it also means that you can spend a lot of time designing experiments and getting ready for them. So how we get to that scale is something that we will try and test before we get to that point. What we do know is that we can get a diffraction experiment where we can get the dilution fridge in there to get cold enough to do that. Whether we can have enough precision in the signal to be able to see those effects, it's outside of my expertise, so I'm just like waffling an answer at this point. But we will do those tests to see what exactly we can do under those conditions. We are actually going to have a town hall. It's a long way to come from California to get to Liverpool, but we will have a town hall over the summer where we will discuss some of these things. And again, if anyone is interested, some of that town hall will be remotely. We can get people on, if you send me an email, we can get you on a mailing list, or to any of these contact pieces of information that are on here. We can get you on the mailing list, and we can tell you how those discussions are going, and we would love to have the input on that.
Okay, thank you. And Nesta says, what analytical spectroscopies are you considering?
So on the Rudy instrument itself, probably very little, because it will be set up primarily as a TEM. And so basic focusing of a beam to be able to do coherent diffraction imaging, but not really to do 4D STEM inside of that microscope. Where we will do the analytical stuff is within our ecosystem. So we will have UEMs that will be support instruments for the Rudy case. And so there we will do any analytical work inside of that. Potentially in the future, you could get an EEL spectrometer on the end of it and do energy filtered imaging. But it might sound like 125 million pounds is a lot of money, but it's not enough to do the development of a spectrometer that would be needed to run at 1.5 million volts as well. So at this point, it will be through the ecosystem or the analytical work.
So I don't know if Nesta has a follow-up with that. But in the meantime, you mentioned about the high voltage being, of course, better for radiolysis problems. But then there's the knock-on damage, and although the electrons are going fast and most of them will go through, then that will be one factor. But on the other hand, are you planning on doing any radiation damage experiments? Who knows if nuclear reactors might become back in vogue?
Well, they are back in vogue in the UK, so yes, is the way that we will do that. So we will. There's also medical applications. People do electron beam irradiation of tumors. So there are medical applications as well. Effectively, we will have a neutron source and an ion source to look at ion displacement reactions and things. Will go through it. In terms of the knock-on damage, yes, it's going to be a problem. We're talking about incredibly low doses. We're going to have doses, even though it sounds like a lot, with 10 to the 6, 10 to the 7 electrons. When they're spread over a 4K CMOS device, or the area that gives rise to those images, it's actually going to be lower than the doses that Hong was talking about in terms of what we will do. And the way we'll overcome that is, again, through the process of using the dictionaries, doing the high resolution, transferring that. So it will be a way of doing radiation damage experiments will be implicit in the process. I think Hua has his hand up as well.
So I was going to say, what are the detectors going to be then? Are they going to be standard CMOS or CCD type?
So it will be a CMOS type, but it will be a little bit thicker. So those work pretty well, and we've already done tests of that and had some studies with the companies that make CMOS devices. I mean, as you know, there's quite a few out there that do this. In June, there is the major microscopy conference in the UK, is actually called MMC, and we will have a town hall at that time to talk about all of the requirements for the detector technologies that go into that, to engage some of the vendors. But at the moment, it looks like a lot of the technology for CMOS that's used on conventional voltages just needs to be modified to be able to fit in with what we will need for Rudy.
It seems like you're going to get scattering in the detector, which could cause problems in terms of location where the electrons were arriving.
There are all kinds of issues like that we have to worry about, and that's part of, again, 7 years is a blessing and a curse. The blessing is that you can go through and model all these things and decide how you want to go through and design your detectors to do that, and that's the process that's underway at the moment.
Okay, good. Wad, do you have any comments concerning the high voltage aspects for biology experiments? Would involve bio to be best used, I suppose.
Yeah, thanks so much for this very comprehensive description of your adventure. So I think you partially answer your questions on the detector. So one of the old idea that both Ken Downing and Bob Glaser had is doing these decelerators before the CMOS detector came into play. So there may be something as an alternative procedure if needed to overcome some of the point that Bob pointed out. So I have another question related to the sample temperatures. I mean, what do you envision the stage temperature? At least you have different types of stages in your instruments. Have you have any in mind as?
No. So we weren't planning on doing anything beyond conventional cryo stages for electron microscopy. The idea was you just have a bigger gap, and so there are things that you can do. Obviously, going into that means that we would have thicker samples for Rudy, which means that we'd have to look at specimen preparation techniques for cryo as well for dealing with thicker samples. And so there's all that part of it. Again, blessing and curse, 7 years gives you a little bit of time to go through and solve some of these problems in terms of the specimen preparation. But at the moment, the plan is to just use conventional cryo stages that are adapted to the bigger size of the microscope. I mean, one of the things that you have for a horizontal microscope is how do you have a cryo that doesn't leak, that you have to figure out what to do with that as well. So all of those, we are starting the process this year. Obviously, we have a town hall on biosciences that you're going to come to in April, and then we will have the MMC meeting, which will be in Manchester at the end of June, where we'll start talking to vendors about what kinds of things could we look at doing, whether it's conventional stages or whether we do something a little bit exotic. And I'd forgotten about the decelerators from Ken and Bob's work, and I'll definitely bring that back up and look into what we can do with that as well.
Okay, that sounds good. Maybe we need to rethink about how to cool the sample rather than the conventional way. So I think with some of the cryogenic technology now in place, it may be a good time to think about that. Okay.
That would be great. I would love to have your input on it, Juan.
Right, good. Look forward to that. Okay, so Mike has a questions.
Right, so we just have this last question. How do you determine when the digital twin will give a different prediction than you'll see in an actual experiment?
Yeah, so good question. Yeah, so I think this is a generic question about digital twins, artificial intelligence, hallucinations, all the things that go into that. I think in the process, people have been doing microscopy for a long time, and we haven't had the highest resolution before, and people still used to solve problems. I think what we tend to want is a one image or one picture solves every problem. We're likely to have to do multiple experiments anyway. We're likely to have to do multiple simulations. We're likely to have to correlate things between the digital twin and the experiment. We don't expect that the digital twin will replace the experiment. We think it's more of a simulation tool that will help make the experiments more efficient and decide what types of experiments we should be doing. And of course, if you get a different answer between your simulation and what you do with the experiment, then there's something that you can begin to test on that. And that will obviously be something that will help us get the digital twin better. But the expectation is that we will have cases where the digital twin won't be perfect to start with, and we'll have to iterate towards the final solution.
Okay. Thank you. So, Hua, I think we should close this formal session now. People have other things to go to. I think you're going to a class. Colin is still in his class, so he can't be here. Hua, do you have any final comments, please?
Okay. I wonder, Hong would care to answer Marine's questions?
Yeah.
If you can unmute.
I think he's asking about potential artifacts into the CTF. Free shot correlation. Maria, don't know much about it, so that's an old topic. If you free shot correlation, if you have artifacts, it's going to have an impact on that. I don't know how that's going to manifest.
Well, I think if you have a sample, you see the feature, and alpha helix should look like alpha helix. So that may be the ground truth. And so there are multiple ways one can look at the feature, but not relying on any single number. So that's another way of thinking about it.
Yeah.
Okay, good. Thank you very much for both speaker, Nigel and Hong. And really appreciate this exciting and stimulating lecture you delivered this morning.
Thank you so much, everybody.
Thank you.
Thank you.
Yeah, Kate, would you like to say anything?
I just wanted to thank both our speakers again. That was a really exciting from both ends. It was great to hear about some of the biological aspects, and Nigel, it's really great to hear about the ultrafast. So two realms of microscopy here. I really enjoyed it. Thank you both.
Good. Thanks, Kate. And so thank you, Hong and Nigel. Great talks. And Jane and Yi for setting it up and running it so perfectly. I think we can finish the recording now and just wind down between ourselves. Thanks.